So this abstract is presenting data for a combination of Ianalumab, which is also known as VACE736, in combination with Ibrutinib. And this was really the phase 1 dose finding cohort that we did to determine the optimal dose for use in this patient population. So VACE736 is an extremely exciting novel antibody. And the reason it's so exciting is that it has two mechanisms of action, and based on the preclinical data, is likely to be highly effective. So the target of VACE736, which is a humanized monoclonal antibody, is the BAFF receptor. So compared to most available therapeutic monoclonal antibodies in CLL that target CD20, this is a completely novel target. And it not only targets BAFF receptor on CLL cells effectively, leading to NK cell-mediated ADCC, it also blocks signaling through the BAFF receptor. So it's a blocking antibody as well as a therapeutic antibody that leads to immune destruction of the target cells. So I guess these two mechanisms can work together to make it really effective. There's preclinical data suggesting that BAFF receptor signaling is an active pro-survival signal that is still ongoing during Ibrutinib treatment, meaning blocking this is potentially effective additionally in killing off CLL cells. And then the activity of the antibody is really outstanding, including ex vivo in the setting of Ibrutinib, where other therapeutic monoclonal antibodies have some decreased efficacy compared to not in the presence of Ibrutinib. So based on these exciting two mechanisms, both blocking signaling through the BAFF receptor and just the extremely potent NK cell-mediated ADCC targeting cells with the BAFF receptor, and the fact that in preclinical testing, this could be combined with Ibrutinib, we thought this would be an optimal antibody to use in combination with Ibrutinib. So of course the anti-CD20s have been used with BTK inhibitors, both Ibrutinib and acalabrutinib, but just based on the mechanism and efficacy, we thought, or preclinical efficacy, we thought this would be a good one to test. And we are targeting a patient population that really has something to gain by the addition of a second therapy, and that's people taking Ibrutinib for at least a year that don't have a complete remission, so still have residual CLL, or people taking Ibrutinib who have developed a mutation known to confer resistance to Ibrutinib, and that would be mutations in BTK or PLC gamma 2. So in these cases, the idea is to add VACE736, which is an antibody, as a second treatment to eliminate the residual CLL, and if patients get a very deep remission, to have them stop taking treatment and be observed ideally. So in order to select the dose, we did a dose-finding study, and that's what we were presenting at the meeting. And we really found that the antibody was extremely tolerable in combination with Ibrutinib. There were some cases of neutropenia, including high-grade neutropenia, but there weren't really infections. The patients did really well. There was no maximum tolerated dose and no dose-limiting toxicities. So we've selected, based on some PK and pharmacodynamic data, and of course the safety data, what the best dose is going forward. And the most exciting part is we did see, even in this small study that was really primarily for dose finding, some evidence of efficacy, where we did have some patients end up having a complete remission with undetectable minimal residual disease at the end that are now on off therapy observation. So it wasn't really designed for efficacy, but it was extremely important to see that some patients did respond and had a decrease in their CLL burden or even achieved those deep responses. So we are now studying this in an expansion to get more safety data and more preliminary efficacy data. But it's just a very exciting, extremely novel antibody.